Hello, welcome to this series, the Toxicity Testing and Safety Pharmacology. Today in this lecture, we will discuss about the safety pharmacology and their various aspects related to the safety pharmacology, why it is important and why it is need to be discussed in this lecture. The safety pharmacology. The word is derived from the safety, second is pharmacology. So here we are discussing about the safety. But my question for you is that which safety and what kind of safety and for what? The safety of drugs or safety of human beings. Safety of human beings with the drugs. This is called the safety pharmacology. Generally, it is defined as a branch of pharmacology dedicated to detecting the potential undesirable pharmacodynamic effects of substances on physiological functions in relation to exposure in the therapeutic range and above. By these definitions, we can analyze and we can understand the role of pharmacodynamic in understanding the safety. And this is telling us about the different receptors and their reciprocations with the drug molecule. Safety pharmacology is vital in the development of new pharmaceuticals. It also ensures that the medications are safe for consumption by identifying any adverse effects on major organ systems or any single organ system. One major aim of safety pharmacology is to predict the potential risks associated with the new drugs before they are introduced to the public and in the market. Thus, it becomes essential practice to perform the safety pharmacology of recently medicines. It is further divided the safety pharmacology in different parameters. First, it is divided on the cellular levels, how a drug is affecting our on cellular levels, then tissue levels, and further is organ levels. Is impairment of organs caused by the drug candidates? So there are certain requirements for monitoring and recording of toxic effects, particularly on three different systems that we have human system. First is the cardiovascular system, respiratory system, and most important, the toxic effects on our nervous systems. How the drug we are taking or administered through the various routes are affecting our three major human systems. They are essentials. If there is slightly change in the potency of a drug or toxicity, definitely our cardiovascular system will be affected. The heartbeat, the hypertensions or many other ailments may occur by slightly change in the drug dose or combinations. In the similar way, the nervous system, as we are aware that no drug can easily penetrate across the blood-brain barrier. The nature has provided our safety parameters and safety guidelines so to protect our nervous system. But certain drugs definitely cause nervous breakdown and nervous neurotoxicity in humans. In cellular toxicity, the toxicity test should be incorporate reports of damage to the genetic materials. When the genetic material is to be tested, this is called a genotoxicity. It means the toxicity is affecting our gene system, they are altering our chromosome systems, the whole genome system is affected by this particular drug or any new drug. So this area of testing is also known as a mutagenicity testing. Now we have two words. First is mutagenicity, second is genotoxicity. The terms are sometimes used interchangeably, though this is not an accurate use of the concept. Why? Let's discuss. While all mutagens are genotoxics, mutagens means any chemical entity, any foreign particles entering in the human body or animal's system and causing gene alterations and gene modification, that is genotoxicity, and the material is known to be genotoxic. But not all genotoxic are mutagenic. Mutagenicity is also confused with the carcinogenicity. Now, we have three different types: mutagenicity, genotoxicity, and carcinogenicity. Genotoxicity is simple when affecting the genes. Now, discuss with the carcinogenicity. Cause of the confusion and why the people are confusing with this concept. carcinogenicity. The carcinogen word is derived from the carcinogen, which means anything which is causing the cancer in the human body. The carcinogenicity refers to causing the cancer or contributing to its etiology. Cancer growth is frequently caused by the damage to the genome, but the cancer growth can also be caused or exacerbated by the disruption to cellular metabolic process. There are the certain drugs they may cause the cancer in our body, like methotrexate, certain hormones, contraceptives. They are also triggering and causing the carcinogenicity in human body. Carcinogenetic testing. Testing for carcinogenic properties of candidate drug is a regulatory requirement in all pharmaceutical trials involving new drugs and nutraceuticals. It is immense importance for human use. Testing for carcinogenicity is hugely complex process, and minimal acceptable process is one short-term study in rodents and a second long-term study in second rodent species. It means for carcinogenicity, we are using at least two minimum species of rodents and different species. First is mouse and second can be a rat. So these two can be used for conducting the carcinogenic activity. Next is mutagenicity testing. Mutagenicity refers to causing a gene mutations. Gene mutations mean altering the gene orders, gene sequencing within the cells, leading to structural changes to the chromosomes. Altering the positions 21 chromosomes, 18 chromosomes on different positions. How we are detecting and how we plan our study using the different models as we studied in further classes. Using the in silico models, vitro and in vivo testings are some of the methods used for the mutagenicity testing, and this is used for conducting the mutagenicity in animals. Teratogenicity. Teratogenicity is a property or the mechanism by which a drug can induce exacerbate the accidents of congenital malformity. Now, what is congenital malformity? Let's take an example. If a baby is born with some congenital disease, it means sometimes a baby having a hole in the heart, or the limb are connected, having a more than 5 fingers, or having born with cut lips. These are the some of the examples of teratogenic. It means a baby born with some abnormality. It lead to the growth retardations, delayed mental development, congenital disorder without structural malformations. With this delayed mental retardations, growth retardations, we have a very good examples. Recently, a movie is launched on this mental health and special children's Sitare Zameen Par. Over that, we can see the certain examples of teratogenic cases. The three risks important for us when and at what durations the drugs are affecting in human embryo system. Particularly three main phases. First is fertilization stage. Second is organogenesis. Third is growth and development term. The fertilization stage starting with the conceptions up to 17 days. The 17 days period, it is known as the fertilizations. Any drug may interfere the gene levels. They interfere with the sperm, interfere with the ova, and can lead a futile egg. After this, 17 days are completed, 18 days is started, and 18 to 15 days is known as organogenesis. It means new organs are formations. By the introduction of certain drugs to the pregnant women or women bearing, expecting a child, during this period, any medicines which is intended for medical health, for any kind of medical interventions may affect the organogenesis. It may lead to the subabnormality, limbs, or any mental retardations, growth and development. This start with the 56 days to terms until the baby is born. The classification of drug is based on animal studies. After performing certain and numerous studies, we validated this phase and the effects of drug at a various level during the child development. Now, the potential teratogens in pharmacy, very common example drugs, ACE inhibitors. Now, the ACE, what is ACE? ACE is angiotensin converting enzymes. It is naturally available in our body. It is released by adrenal glands. The adrenal glands, it is placed or located above the kidney, and the function is to release angiotensin converting enzymes. When the human body is suffering from the low blood pressures, the nature had given glands, the adrenal gland will release the angiotensin converting enzyme. It will release the renin and it will increase and help in the regulation of blood pressure. But sometimes its over excretions can decrease the blood pressures, and for that purpose we have a certain inhibitors known as ACE inhibitors. Example, lisinopril cause teratogens in females. Acne medications. Acne medications is very common during pregnancy, and certain people recommend isotretinoin. Isotretinoin is a derivative of vitamin A. Alcohol consumptions, some metabolic androgens, antibiotic, particularly tetracycline and doxycycline. Tetracycline become important as when it is given with the calcium products, it may cause certain disorder and malform the teeth in the fetus. Anticoagulants, example warfarin. These are the classical examples of potential teratogens in pharmaceutical products. Others include phenytoin, valproate, carbamazepine, lithium, and anti-cancer therapy. The drug most commonly used frequently and also known as anti-metabolite class, methotrexate. Methotrexate is causing teratogens in ladies. Anti-rheumatic chelators, penicillamine, antithyroid agents, propylthiouracils, and carbamazepine. Certain contraceptive hormones or de-estrogens, example diethylstilbestrols, very commonly used and easily available in the market. Doctors are prescribing these hormones, and this is also causing the teratogenicity in populations. The cocaine addictions, and as we are aware, the thalidomide disasters that lead to the phocomelia in offsprings. The teratogeny testings must be carried out under International Council for Harmonizations of Technical Requirement for Pharmaceuticals for Human Use Protocols. Whenever we are planning this study, teratogenic study, it must follow ICH guidelines and must adopt ICH principles and guidelines. They include the ICH protocols include the fertility assessment, the postnatal evaluations, pregnancy stage susceptibility, developmental toxicity and assessment. These 5 parameters need to be done during the teratogenic testings. Fertility assessment of new species, postnatal evaluations, the birth is baby is born, so we need to check the postnatal evaluations, the head circumference, the voice, the noise, and certain behavioral parameters can be checked after the postnatals or after birth. Pregnancy state susceptibility, development toxicity assessment. Now, the safety pharmacology testings, 3 major organ systems. First is CNS, respiratory and cardiovascular systems. In CNS, we normally check the motor activity, example ataxia and dyskinesia, whether the movement is regulated or not. Like we are able to work or to perform our activity with ourselves, or do we need any support from the family members or from the nurse or medical or any supporting medical staffs. And kinesia is a dyskinesia, the movement disorder. We are unable to hold a glass of waters. We are unable to move upward, downwards. We need some support system. In That's why we need to understand the role of the certain drugs affecting our central nervous systems. Behavioral. After taking some medications, we feel some emotional liabilities, some agitations, we feel irritations. Coordinations, we are not able to coordinate with a certain times with our body. These are the certain side effects of the medication on our CNS. The body temperature is sometimes is very low, sometimes is very high, or some drugs is raising the body temperature as they are affecting directly to the hypothalamus. The hypothalamus is disturbed and they are raising the body temperature. These are the some major side effects of drugs affecting the nervous system. In respiratory system, what they can do? Mostly they alter the respiratory rate. Either they increase the respiratory rate or decrease. Tidal volume and oxygen saturations is three major parameters need to be done and to be evaluated for respiratory toxicity. Now, third, the cardiovascular toxicity, blood pressures and heart rates. Is one of the most convenient way to check the blood pressure after taking the medications, and you can perform at your home using some apparatus. While you are taking some medications, you can start the BP measurements using clinical apparatus or at your places. So this can correlate the effect of your drug at your body and heart rate. The ECG, repolarizations, QT intervals, tossets, the pointers. The HERG assays, potassium channel screening, these are the some parameter needs to be done while a drug is administered within to the animals or to the human system. Thus, the safety pharmacology developed after the tragic incident of terfenadine. The terfenadine problem was identified in 1990s. Although the risk from the drug toxicity had been known for many decades, apart from certain drugs, they are also causing some side effects in human system. First is sulfanylamide. Sulfanylamide is collaborated with the some ethylene glycol, and the pharmacist, chemist, druggist, they collaborate and formulate the elixirs. Elixirs cause certain disorders and this lead to the deaths of 50 plus children. A thalidomide disasters. Thalidomide is used for the motion sickness, and this indication is used by the pregnant lady as during the pregnancy it is very common the morning sickness. The women pregnant lady feel some vomiting or some difficulty during the morning time. So to avoid this, the many doctors prescribe the thalidomides. This drug show a wonderful effect in sickness, morning sickness. But after administering these drugs, this is affecting the fetus. It means teratogenic effect. The newborn baby born after the administration of this drug in certain areas of Europe lead to the formation of phocomelia. Phocomelia means the newborn baby born with fused limb systems and fused hand systems. Other tragic drug is minoxidil and third generation of oral contraceptives. This is the key incident that lead to the safety pharmacology. Now coming to the terfenadine. Why terfenadine is important? Terfenadine is a non-sedating and antihistamine. It means antihistamine is given when there is an allergic condition in our body. Before this, whenever any histamine is given to human body had severe ADRs, adverse drug reactions, particularly associated with sedations and psychomotor retardations. It means after administrations of terfenadine, The people suffering with the sedations and there is slow activity of psychomotor. Hay fever sufferers were faced with the effects of the disease or the debilitating effects of the ADS. Now, what is hay fever? Hay fever is a allergic rhinitis. It is not an infection or any disease, but it is generally caused by the certain pollutants and causing continuously sneezing. The earlier antihistamines cross the blood-brain barrier. It means when any drug is crossing blood-brain barriers may harm and may cause certain and severe side effect to the nervous system. This problem is resolved by the terfenadines. It as first is it is non-sedating and third is antihistaminic. Terfenadine, an active metabolite, did not cross the blood-brain barrier and therefore did not cause the drowsiness. No sleep, no slowing of thoughts and movement hay fever patient often complain of. The problem faced by the patients is resolved by the introduction of terfenadine. So, terfenadine was so successful and popular, it became an over-the-counter medications. The people are purchasing for allergic, for hay fever. Terfenadine become a very popular in 90s or 95. But later on, the problem is identified by certain doctors. What happened with this? This prolonged QT interval and causing ventricular tachycardia. Tachycardia caused by the terfenadine. Next is exacerbated in patient taking other medication that down-regulated cytochrome CP3A4. This is a enzymes and it is down-regulated by the terfenadines. Terfenadine was metabolized by cytochrome A3A4. By the 1997, a major incident took place. In this incident, 14 deaths reported in United Kingdoms and the drug was banned and withdrawn immediately. It had taken 10 years for the cardiotoxicity danger to be determined. How much time? The 10 years. The scientists, the pharmaceutical scientists, others, toxicologists took 10 years to focus on the terfenadine toxicity. The drug safety pharmacology testing, including the EGC testing, was now established as a standard toxicity study requirement. Now, the safety pharmacology focus on three major aspects. First is nervous, cardiovascular, and respiratory systems. So, this is a major incident in the history that lead to the establishment of safety pharmacology and lay a foundation. Toxicity testing is essential to save the lives and prevent the injury, as we noticed and we experienced in thalidomide and terfenadine. To ensure the pharmaceutical companies do not waste time and money on researching a non-marketable products. It is useless and unessential to research on a product that is already not available in the market and to do more research on that products that is not a gainful insight. To avoid hugely costly litigation if the drug goes on to kill or injure the patients. The toxicity testing is a regulatory and legal requirement in all drug development in all phases. Pharmaceutical products cannot be marketed unless they are approved and licensed. State regulatory bodies will scrutinize toxicological studies before licensing a product. Animal human equivalents. Animal differs from the humans in numerous ways. First is structural, second is physiological, third is biochemical, fourth is genetical, sixth is sentience. These are the parameters that differ the human with the animals. During the animal studies, these parameters cannot be evaluated. That's why it is essential for a drug to be monitored and to be under vigilance for long-term toxicity, and this is established by the terfenadine. The animal test failures. This is the live example of theralizumab. Theralizumab TGN1412 is a monoclonal antibody and known as a human monoclonal antibody designed as a treatment for chronic lymphocytic leukemia, the blood cancer and rheumatoid arthritis. It is a great success for against the blood cancer and rheumatoid arthritis autoimmune disorders. Bind to immune system T cells at receptor CD28. The drug was first trialed in humans in 2006, nearly 20 years ago. 6 health volunteers suffered catastrophic organ dysfunctions even at subclinical doses. How? While the drug is approved from the preclinical research, there is no such cases were reported. And a small dose, not a subtherapeutic, less than the subtherapeutic dose, and 6 volunteers face a severe side effects. The human clinical trial had been preceded by testing on a non-human primates with no apparent ADR of that nature, and non-human primates do not express the CD28 receptors. These major incidents in the history lead to the safety pharmacology. Thank you.